thank you very much. So I'm Mickael Baque. I'm French, but I'm working in Berlin at the German Aerospace Center at the DLR. And I'm going to talk about how to find life on Mars, as you heard in the song. But to do that, we exposed some samples in space on the BIOMEX experiments on the XPOSE-R2 platform. And I've been looking at preservation of Raman biosignatures from different organisms after space exposure. So as you may know, we have been to Mars in the 1976 with the Viking landers to really search for life there. And so it's been 40 years since we really sent dedicated missions to search for life on Mars. And a few days ago, it was also 40 years since the first Star Wars was released. I don't know if it's a coincidence or not. But so we are almost ready to go back with new tools to really search for biosignatures, as we heard, those things that we can think were made by life on Mars. And one of the tools that will be doing this job will be Raman spectrometers, or we hope so. So the next two missions, the next two rovers to Mars, the ExoMars rover 2020 and the Mars 2020 rover from NASA, will carry Raman instruments to Mars. But as we learned from the Viking missions, we had to understand much more about life on Earth before we can find it elsewhere. And so that's one of the goals of this BIOMEX experiment that we sent into space. And so we are looking at lots of different microorganisms from Mars analog environments on Earth. And after exposing them to different conditions, and especially simulated Mars conditions, to see what we can detect after this exposure, what we can expect to detect with such instruments, such Raman instruments on Mars. So to simulate, of course, Martian conditions, we can either use ground-based facilities like Mars chambers to simulate radiations and so on. But of course, when you want all the combined effects of Martian environment stresses, you can go to space. And so there have been a lot of not a lot, but a few platforms to do these jobs. So the Biopan platforms, or first also on the space shuttle, or the long duration exposure facility of NASA. And but at the European Space Agency, we've been doing with the XPOSE platform. So we had 3 missions so far on the XPOSE E, R, and R2, the last one. And so I had the chance to be involved in this mission during my PhD and now during my postdoc to analyze the results and the samples that came back. So this mission was launched in 2014 in July. It was installed on the outside of the International Space Station in August on the Russian segment, so that's why it's called XPOSE-R. And so it began exposure in October. We removed the cover and so on, and the samples came back to Earth June, March and June. So after 16 months in low Earth orbit, so in the space environment of the space station, outside of the space station. So it's not exactly like you would find on Mars, but you have still all the cosmic ray radiations, the UVs. We are simulating all of that. And so there were 3 biological experiments on XPOSE and 1 astrochemical experiments. And among the biology were the BIOMEX experiments led by my supervisor at DLR in Berlin. So that stands for Biology and Mars Experiment. And as I said, the big goal was to look at the stability and detection of biomolecules under these Mars-like conditions and space conditions in order to create a biosignature database for the future Mars mission to really try to guide these missions to see what we can expect to find from all the examples we have on Earth. And also as a byproduct, of course, because we are exposing organisms, we want to see the endurance of these different extremophiles under these conditions to assess the habitability of Mars, the lithopanspermia theory, and also we have some kombucha samples that might be useful to support the immune system of astronauts, and also, of course, to test for future space exploration instruments. So we had, it's a big group in BIOMEX, so we had 27 collaborating institutes from all around the world, actually. And so we were able to expose 15 different organisms from the 3 domain of life, and 9 different biomolecules, so isolated molecules from these organisms or others. And we mixed them also with 2 Mars regolith simulants and 1 lunar analog to see also what is the the interplay between the minerals and the organisms, or the minerals and the molecules, to see if there can be better preserved in certain minerals, or if they can enhance the destruction of the molecules in other cases. And just a bit of technical details for the mission. So we had 3 trays on expose. BIOMEX was here in the red part. So the first tray was the space, so it was fully evacuated to space vacuum. And it had magnesium fluoride windows to have the full UV radiation from the sun. And the second tray, which was filled with the Mars-like atmosphere, so Mars-like gas at 6 millibar pressure and CO2 mainly. And it had a cutoff filter for the UVs at 200 nanometers to recreate Martian atmosphere conditions. And we had also a top and the bottom layers to have the fully exposed samples, and the bottom to serve as a dark control in space to see what is the difference between UV radiations or the samples only irradiated with the cosmic and ionizing radiations from the sun and so on. And at the same time, we had also experiments on the ground to serve as a control, also on the ground, to serve as a reference samples. So here, of course, we cannot recreate the full ionizing radiation spectrum. That's why we go to space. But we can recreate mostly the UV radiation, vacuum, and Martian temperature, and then Martian atmosphere, and also temperature cycles that the samples are in parallel experiencing into space. So what I was looking at mainly in the last few months were carotenoid molecules comprised in cyanobacteria and green algae. So carotenoids are a very wide variety of molecules present in many different organisms, mostly photosynthetics, because they act as secondary pigments for photosynthesis. But they have also lots of different functions, like they could be some prebiotic lipids also of interest, but they are also very powerful antioxidant molecules, so they are found in lots of extremophiles, organisms that live in very harsh conditions and that have to deal with lots of stresses, and so these molecules are very powerful to scavenge reactive oxygen species, for instance. And also what is very interesting is that they have a very high preservation potential. Like you can detect some carotenoids by Raman in 1.4 million years old samples, and even the byproducts in 1 billion years old samples by GCMS. So it's quite a good molecule. And by Raman, yes, it has a very distinct Raman spectra, so it could be very good to detect them on Mars, and potential Martian biosignature. So these two organisms, yes, so we had a nostoc cyanobacteria from Antarctica and spherocystis green algae from Spitsbergen, from Svalbard, and so they can go into resting stage and accumulate these carotenoids, as you see this orange reddish color there. And so we prepared, this was done by our colleague Thomas Leya in Potsdam. So we prepared them, as I said, in contact with these Martian mineral analogs to see what is the interplay between the minerals and the cells, and if we can protect or destroy more the biosignatures. And so they were plated on these different analogs, and as you can see under the microscope here you have the big colonies of nostoc, and all the little dots here are the spherocystis green algae cells. And the first result, of course, is that they survived. After 16 months into space, they came back, and our colleague Thomas Leya put them in culture, and they were growing again. No problem for them, so that's already quite a result. Not so surprising, because as I said in the previous missions, we've seen that lots of different organisms can survive space exposure. So we have seen from other cyanobacteria that they can survive after 16 months or even longer into space. Even some green algae also have been already exposed. Tardigrades, the most famous water bears, as the password, yeah. But so we have already some examples of organisms that survive. But still, when you add more and more that can survive this condition, it's already good. And so what I've been doing specifically, it's looking at them by Raman spectroscopy. So looking at, if you don't know Raman, it's inelastic scattered light that is created by excitation of monochromatic light on the samples, and you have this fingerprint of the different molecules that are very distinct. And so it can be used for biomolecules, but mostly also for mineralogy. So it's a very good for Mars to have the mineral context, but also if you have a biomolecule there, you could detect it. And what can we see with cyanobacteria and green algae? So this is the typical Raman spectra of carotenoids. So you have the free peaks with the vibrations of the carbon-carbon liaisons and carbon-methyl liaisons. But what we have in these organisms are not, of course, single molecules, but a pool of all the different carotenoids expressed in them. So this spectra actually is not from one molecule, but from the whole pool of the molecules. And so we can do different images that or different line scans to see the intensity, or draw maps from the samples here from the nostoc colony. So you can do the intensity map of the carotenoid signal on them. And these are the results after space exposure. So as I said, we have all the different minerals here, and here we have the top and bottom positions. So these are the intensity maps. So the first thing you can see is that you have signal everywhere, even after 16 months into space. So it's not really surprising because they survived, so it shouldn't be completely destroyed because they can even grow after that, but still the signal is completely preserved. And if you take the average of the spectra, you have almost no difference in the spectra of all these conditions, either on the different minerals or without minerals at all. So that's already quite a promising result. But yeah, we try to go a bit into more details to see if we can have if we can quantify this preservation. And this is still very preliminary. I need to have students to code for me because I'm not very good at that, to analyze all this data. But what we can see is the intensity maximum of the signal that we obtain from the Raman spectra. And here, for the moment, it's a bit rough, but we still see no difference for the nostoc samples in the maximum intensity, more or less. But it's a bit different for the spherocystis, so for the green algae. So the green algae are eukaryotes, so more advanced organisms. And first of all, it's a bit more difficult to spot them when they are mixed with minerals because, of course, you cannot see the big colonies of nostoc on these samples, so the coverage may vary. So you can pick randomly some places to analyze, but it's not the same everywhere. And here we have some differences between the different minerals used, or without minerals, and the different minerals used, as we have a huge fluorescence coming up. So I didn't say, but we have a green laser, so that's the same as I'm using here. And so that's a problem for photosynthesis, for biological molecules to analyze with a visible laser, because you produce a lot of fluorescence out of it. So that could be a problem for Mars too. And here it's a bit messy, and we have a lot of differences, but we still need to go a bit more into details about that, and to see the trends between without minerals and with the minerals, what are the effects that they have. But it seems that the minerals can protect a bit more than without the minerals here. So that's that's for the moment, that's what we have. So there are very promising biosignatures that we can detect by Raman, and hopefully on Mars in the future. And we have also statistical method under assessments. And we have, as I showed you, many different organisms also containing carotenoids. So we will compare with all of them and see what we can say for a future database, and hopefully what we can detect on Mars in the future. Thank you.
Which kind of media you use to grow your bacteria?
So the nostoc, they are on BG11, the typical Cyanobacteria medium.
Okay, so in that experiments you study the physical effect of physical parameters on your bacteria. You not mimic the mass environment chemical properties in your media, right?
No, not so much.
Okay.
We just have the minerals mixed with that, but yeah. So
okay,
some other,
You use the same concentration of the chemicals also, which are present on the mass in your media?
No. Just the mineral analogs are analog mixture minerals of what we can find on Mars, but no, with the medium and so on, no. So just to see really the connections, if there are some small effects.
Is there any change with the growth rates and the growth pattern of your bacteria in space?
No. They are completely desiccated and so on. So when they come back, like we put them in normal liquid medium, or we plate them and so on. And so no, we dilute them and plate them. So there is no influence on that. Yeah.
Thank you. It's interesting, is it?
I wonder if you've looked, or if anyone's looked at transcriptomics before and after sending into space to see if their gene expression changes.
Yeah, no. We send these samples for single-cell DNA analysis, but that's not transcriptomics. I don't know why they are doing that. Okay. But no, they are in desiccated form, so that's just going to be repair mechanisms activated after when you put them back. So these are, maybe you will have you can simulate this on the ground. It's not, you can accumulate damage, and that's just the capacity of the cells to have enough repair mechanisms. They can repair all these damages, but that's what they are experiencing anyway already on Earth, in Antarctica and so on. They have to. So that's what they are good at. Yeah.
What makes Raman spectroscopy special in your case? Is it because you have some symmetrical molecules, like they all have center of inversion, I think?
Yeah, these carotenoids, because they are pigments, so they absorb and they in the visible spectrum. So when you use a visible laser spectrum, they have a resonance effect. So you enhance the Raman signal a lot, and so you can detect them very easily. So that's why these molecules are model molecules for Raman for biosignatures, and then, the good thing about Raman is that it's a non-destructive method, so you can use very low laser powers and so on, and you're not destroying them. You're not destroying the molecules, so then you can use another technique to confirm. As we heard, we need a lot of evidences to make an assumption about the biosignature, really. So you can have a first glance with that, and then use another technique to prove that it's really there or not.
All right, thank you very much.
Thank you.